just like they just don't show up for office hours.
But have we met? We probably haven't. I have. I'm like, I'm sure.
I have like a week.
Computer
2
programmer
weeks
and
that have no
Oh, good.
job, and the other weeks have
Okay,
only
I don't
one
know.
people show up. And last week,
to know
2
microimmunology.
people show up.
What are these pockets?
Are you?
One of them is more
you a
like
computer
asking
programmer?
less homework questions, and one of them
Good.
is asking
Ah!
That's what I do. That's what she hires
You have one in there, but they actually
Yeah, I
inside
tried.
is.
Oh, it's
Well,
like
just
it's
for
so-
one module.
My first
Like, if
exposure
I
was
need
the
to-
IBM
It has been
360.
split into
grab
3
something,
modules.
Manipulate
or
something.
write, or yeah, open
Fortran.
the door, I can
What is
use
it?
those.
I don't even need to create the homework assignment.
I really like them.
Give
They
Steve
just
be like, I'll take
And
care
I'm
of
in,
this.
and I can write or anything.
No,
I
my
can do
son-
field work at the
Well, my
time.
son's in the
So
computer
that-
science.
I mean, all you did was
grab something.
all you did was change it to all
On
your.
the keyboard.
But now I use them to put my.
So the same link will give you the same.
Yes.
Oh, I guess they do.
Now I use them to handle.
I don't
You
think
want
they're
to talk
changing.
later today? It's gonna snow like crazy tomorrow. I don't have.
I guess Swami.
Yeah, Steve. What about the? Aren't there graduate students that are being
Yeah, I
interviewed?
really like them.
I think
About some stuff.
tomorrow's the interview day.
Yeah,
Yeah,
I
but
think
tomorrow's
I
gonna
kicked out of
snow a
that
lot.
one.
Right?
I think so.
And a lot. Delta cancel all its flights.
They'll make their choices.
I don't have enough lycra.
You'll drive them to choose another place. You mean?
It's not major, so
Yeah,
yeah.
I'm not
I have a lot of.
And let us know it's hardy enough to be here.
Oh, so if they're playing it and they don't, what? How are they going to come otherwise?
You find a way. You get in the car.
Even if you're coming from.
Call out the Husky Brigade.
Yeah, I'm lazy. I still don't have those yet.
Oh.
But snow season's almost over.
Yeah, you don't need it.
Yeah, I just think it's let's start.
Take a dog sled in.
That's easy.
No.
Oh, they
Yeah,
have.
okay, right. What are we?
They're just like.
actually.
I was happy that you ran and it worked.
That you said?
I don't sign.
Nobody really totaled it. I think it's
I mean,
for
do they look
auditing
to check? Oh.
It's for auditing purposes.
What is that?
So
The
it
untrained people
justifies
actually take
whenever
hold.
you do
Oh, I
anything
see.
on campus where you're food.
Well, they do it for
Yeah.
seminars,
Yeah.
environmental seminars too.
Like,
There's food. I think they got
let's do rolling this.
Oh, I see.
I already.
That makes
Where you
sense.
do food, you have to
They-
put
they-
it
they-
in
one
the
way or
There's
the
a
other.
lot of like seminars and they don't
faculty
ever take names. Or
Yeah. It's definitely food involved usually.
No.
Ones
Yeah.
I've
So
have been nicer than.
I thought it was like Brian or Linda were
yeah.
noting who's there and who's not.
It's they don't want people who have access to accounts to buy food, take it home,
Yeah.
and waste money or buy.
Who would actually? I don't know. I can't imagine
There's always
doing that.
somebody would do that.
Alright, so we're going to go ahead and get started. We got a full house, so that's great. Welcome everyone, the Tools and Technology Seminar Series. I think you've all been here before, so I don't think I really need to go through a big explanation. And since Brian has walked in, if you want, you can introduce
Well, boy, we got a big crowd here. That's good. I'm sure that I can do it if you'd like. Sure. This is a timely talk. You know, this. I'm very curious to hear what Gerry's got to say since he's got so much experience in the industry. In this, like everything else in society today, this AI word, certainly overhyped, is you know, taking over the frontal cortex of some of our leadership. Various levels that want to know what we're going to do about AI and this and AI and that, and AI, you know, putting their shoes on in the morning. So I think it'll be fun to hear from Gerry. I've worked with him for many, many years. He's a research professor in the department, works embedded in my laboratory, and you know, he's got a lot of background. He was a section chief at the NIH, I think the youngest one ever. Has been a pharmaceutical company technical executive at various places, Roche, Merck, and so on. And has worked in a bunch of companies over the years, including our latest one that we worked together on, Assurance Health. And you know, and just you know, applying the regulome to pharmacoepigenomics. So go ahead, Gerry.
Okay, thank you,
You
Brian.
had something to eat.
Yeah. Although So, yeah, I was going to sit down, but, that's probably not appropriate. So, this is not really a tools and technology talk. So, anyone that came for that can leave. So, okay. No, serious. This is more a overview and applications talk. I meant to do this because, for some reason, out of desperation, I was asked to give workshops at the end of this month and at BioData West, which is next month in San Francisco, on kind of the foundations of artificial intelligence as applied to drug discovery this month, and next month is more about the regulome and AI. So I thought, well, good, I'll force myself to learn something about this domain, right?
By the way, I can relate to this. I had to get to the molecular health board on, hey, what do you want to do about AI, doctor? I mean, people get called in to do this. Oh, I know. I mean, it forces you to think.
Yeah, but I didn't think too hard about this talk. But anyway, so I push a button. So I'm going to talk a fair amount about drug discovery, and it's sort of the data associated with that. A little bit of small piece about competitive space and growth rate, and then very brief examples of two applications. And people are welcome to ask questions, although obviously if we get bogged down for half an hour, we'll never get anywhere, but that's fine too. So here's just some definitions from Wikipedia. Machine learning, deep learning, unsupervised versus supervised. Unsupervised is where the data are unlabeled. Do people all know this? I assumed in this department people would know sort of what these were generically. And I'm going to just touch on a brief collaboration that I had with a fellow at MIT, Prakash, on probabilistic computing, using one of his approaches, or one of his pieces of software called BaseDB. Okay. So this is from a deck from last year's BioData West, courtesy of Jeff Dean at Google Brain. And this is sort of the what was going on in the 80s and 90s, which I can relate to because I was at Roche. One of the things I was overseeing was something in Roche image analysis systems where we were making an automated pap smear image recognition system, and for digital pathology, if that makes sense, to try to find cancerous cells in a pap smear. And at that point, this has a lot of resonance for me because neural networks weren't really sophisticated enough to be able to operate like they're doing now. And we had to use algorithmic approaches, and we could never equal the accuracy of an actual pathologist looking through a microscope, which is sort of the standard you have to meet or exceed for FDA approval, right? So anyway, now that's no longer the case, although, so it's accuracy on the y-axis, scale, data size, model size on the x-axis, but of course that requires more compute time. And I like this slide because it's kind of reinvigorates my interest in BaseDB and probabilistic computing, which some people here may know about or not. This is again from Jeff Dean, who, by the way, is, well, I shouldn't even, I'm about to make a political statement. I shouldn't do that here. But let's just say some of the people from our computer science engineering program are going out to his group for fellowships and such, you know. So obviously, machine learning makes computers that are software that's able to learn. And his point here, I think, was that actually programming software or a computer to be clever is harder than programming a computer to learn to be clever, or software to learn to be clever. And so this is more falls into the realm of what I'm going to talk at the end, which is a little tiny bit about probabilistic computing. Now, this is my somewhat inaccurate history of drug discovery. So I don't know if you folks, probably Jeff knows about this, and will correct me, I'm sure, without any hesitation. But there are a lot of serum-based stuff prior to about 1910, and vaccines. So all these things, just because I have arrows up here, doesn't mean they're not continuing over time. Obviously, right? Polio vaccine wasn't until later, but this whole period of so-called rational drug design, we sort of had some notion that you had a target and at least a simple mechanism, and you would use that target and design a small compound around that. And that proved to be very good until it stopped working because there were no new drugs and kind of productivity and everything fell. And you can give me feedback if you... Of course, when the human genome was sequenced, there was all this wonderful optimism about, okay, now we know genes, so we can do a single gene to drug screening approach. And this is sort of the era when monoclonal antibodies, drugs that end in AB, these guys, and then the other drugs that end in IB, which are intracellular inhibitors. Sorry. They began to emerge. I'm going to talk a little bit about these two in a bit. And then there's been a focus more recently on gifts of nature. That is large familial pedigrees in which you find the pedigree itself is somewhat rare, but you can sort of identify what the molecular problem is and then you can figure out maybe this is a druggable mechanism or target. That makes sense. And so this is kind of target-based therapeutics. I'm going to focus on this, which is molecular mechanism clinical indication-based target pairs, which is these are kind of buzzwordy things in pharma. And so we're starting to enter the era of deep learning for drug discovery, which I think Alex K back there says, what is really been discovered yet? Which is a good question to put out there. And then maybe finally intelligent machines or real artificial intelligence, like the last slide, where the software and computers are sort of autonomous in some manner.
I guess wearables would be part of that.
Yeah, wearables, and they're on a coming slide too.
Physiological measurements.
Right.
Yeah.
It's more automated. Just a couple of things down here. Companion diagnostics were really touted at the early part of this century, but they have not really flourished like the FDA had hoped they would. But it's starting around 2013, they become more common, especially in oncology. And of course, Brian Apey and I have been involved with a company that was doing pharmacogenomic diagnostics in psychiatry, which was quite successful. Assurex, now part of Myriad Genetics. So any questions, or I know this is inaccurate. If you want to.
It's a great slide, really.
I think you could add a couple of things when I talked with Ray Rudman about some of the traditional methods, art, intuition, right?
Intuit, yeah.
That's
Intuitive
how he talks.
Yeah, well, I thought there's a lot of truth in that, actually. But so, if you do use machine learning or deep learning, which is a subset of it, what data should be used as a training set? So I'm going to spend some time on this because this is a somewhat onerous challenge for pharmaceutical drug discovery. And I guess you're aware, or you should all be aware, that we now have things in more broader landscape of healthcare that can do, especially based deep learning applications, that can perform digital radiology better than a pathologist, and can do some digital radiology better than a radiologist. Of course, those clinical specialties have not agreed with this necessarily, but it's been shown pretty objectively. Okay, so
Also ophthalmology.
in ophthalmology, yeah. So there was this original sort of rational drug design focused often around catalytic pockets and enzymes, receptors, drug transporters. And then there's kind of a switch to, well, we now have genes, so maybe we could just design drugs around single genes that might have some kind of consequence. But most of those failed in clinical trials. John Pritchard and his buddies came up with this omnigenic model, which was published quite recently, suggesting that there were core genes that should be used as, or their products should be used as targets, but there are a lot of peripheral genes in which variants had relatively small effect sizes. I guess you've seen these, I don't know if you've ever seen these XY charts with 6 balloons or spheres in them. And
Yeah, well, Mark
Mendelian,
McCarthy showed one that we looked at the other day.
yeah, Mendelian disorders that are large effect size and rare. Well, in work done by R. Allen Foyer, looking at his computer, and a paper we published, as well as Lillian Wang, have shown that actually pharmacogenomic SNP effect sizes from genome-wide association studies are pretty large compared to disease risk SNPs. And you know, SNPs are not just necessarily a single base change, they can be a small insertion or deletion. At least according to definition, I think it's still active from NCBI, or not NCBI, but National Library of Medicine.
Jerry, let me throw in another one for you, everybody, and you especially to consider here. Is that Xiaoming Wang and some of his new company, and especially in oncology, to avoid the resistance that one drug might have, you throw a cocktail of various compounds against this pathway.
I'm going to talk about that.
Oh,
actually.
good.
Not about his work specifically, but about this approach.
I think that's an approach that lends itself to us.
If you look at this, that's really what I'm talking about, molecular network therapeutics. So you're drugging various parts of a molecular pathway, if that makes sense.
Yeah, good.
Then David Batchelor, who was at the Broad Institute, found that if you look at these natural gifts that occur in nature, again, these familial pedigrees, you can find these families, sort of, I go back to when I was funded by the Hereditary Disease Foundation and Huntington's disease, Nancy Wexler and her father, where they used, I think, pedigree from Lake Maribor or someplace like that in South America that had familial Huntington's disease, which is how they finally discovered what I think the cause was. I know they did, although I don't think it's yet druggable. Anyway, the point being, he showed that for the product of the gene SCN9, that there are various kinds of pain disorders associated with mutation in that gene, thus making a good candidate for drug development. And I'm going to go on to this a little more detail. Then we have work from originally from Josh Denny and collaborators at Vanderbilt, using phenome-wide association studies in which they've identified, of course at Vanderbilt they have BioView. I don't know if that's still extant. Do you, Brian? I think it's replaced by Epic, isn't it there?
They still have the backwardly archived BioView, and they still are populating BioView with Clarity. Yeah.
Okay. So one of the things that came out of this is that they showed that drug response and disease risk variants often cluster within the same regulatory pathways. And we've also shown this with lithium in a paper we published. And then even more recently, a lot of big pharma, and I'm going to go into this in some detail, have formed these relationships outside of the United States, some inside the United States, but mostly with other countries, where they can access a combination of clinical data from like an electronic health record on patients, as well as genotype data or even genomic sequence data or expression data, which in the US is hard to because of our privacy laws. So anyway, I'm going to talk about this a little bit. And then finally, I'll show one sort of slightly inept study, but anyway, where Zoe et al. Published in 2009 what they predicted when they looked at drugs that were already in phase 3 trials would be eventually approved and thus successful, and those which would not. And then they, of course, published a follow-up paper showing that their predictions in silico using machine learning were very successful. Anyway, I'll show you that as well. Any questions? Okay. Maybe this will be over really quickly. So this is from a talk I gave at Teva the day the CEO was fired. It's interesting. Did I tell you about this? They took over Cephalon, which is a company started by Frank Baldino, who's now dead, but was a kind of a colleague of mine. And so their U.S. headquarters is in Westchester, Pennsylvania. And Teva, I think they're the largest. Their headquarters are in Israel, but they're the largest generic drug company in the world. And by the way, it's pronounced Teva, not Tiva. So if you go get pills and the pharmacist says, "Do you want Tiva?" They get really mad about this. Anyway, so what is interesting is he found these families that had these different mutations, and this is very severe pain. If you look, I don't know if you've ever gone to the doctor and seen, well, they have this chart of 1 to 10, and then they'll have faces that go from really bright to smiling.
This is actually turning out to be a big deal, and we've got researchers in Midas looking at this. And if you really want to get blown away, log on to the real-time Twitter feed of emojis that's out there on the internet. It'll shock you.
Yeah, these are very primitive emojis.
These are actually emojis are going to turn out to be very important in reporting things back into the for healthcare research or stuff like that from people. Yeah.
So he found these familial pedigrees, and this is severe pain, and your skin turns red and stuff. This is not as severe, so you can look at the function of this channel, which is encoded by the SCN9A gene, and then look at the pain phenotype. And it's all good, no pain disorder, wild type here. And why this person isn't smiling is because they're actually insensitive to pain, which is not a good thing to be. So actually they should be frowning. But anyway, so he found this, and this is sort of the era of the gifts of nature, family pedigree genes, finding variants, and then developing drugs around them. So let me go another slide. So this is the SCN9A gene, and there are at least 2 drugs in phase 3, raxetrogene, I don't know if that's the right way to pronounce it. And Jeff, who comes up with these drug names, do you know? There must be a brand.
Marketing guys.
Marketing guys.
I don't know how they come up with like a Abilify.
They also have to have letters that make them so they won't be confused with things, and they have to.
I know that, right? So the pharmacist won't give you the Schedule 2 opiate instead of the
they have to go through
aspirin or whatever.
Trademark office, or so they don't get sued.
Oh, for crying out loud! Anyway, so this for severe fibromyalgia, which actually has a different clinical name now. This is a Mexican female pedigree. Sorry, go ahead, Steve. See if Rory saw this plan. I'm kidding. He thought he was going to be really. So that was kind of neat because these show the promise, although they're not yet FDA approved. So that, moving forward. So a lot of big pharma, I'm going to get there. You can see some of the partnerships down here between various companies and some of the, this is the U.S. Geisinger Health System, as people may know, is a very progressive health group. It's in Pennsylvania, isn't it? They do a lot of genotyping and stuff. Regeneron has worked with them, but Roche is now working with Estonia Electronic Health Record. Teva's worked with Finland. Amgen with Decode. So these are sort of the drugs that have are either in phase 3 or approved. And of course, to me, what's interesting, because I wrote a paper with Hardy about the amyloid cascade hypothesis in '92, is it looks like the new generation, the very newest ones, are effective. And at least one I know has just received breakthrough status, in Alzheimer's disease, late onset. So for me, that's kind of neat, anyway. And for anyone that has Alzheimer's disease in the room, it might be a good idea. But like me. So this is that paper from, there's a 2009 paper from Zhu et al., I hope I'm pronouncing correctly, and then a follow-up paper in Trends in Pharmacological Sciences in 2017. And they used machine learning back then, support vector machines of DNA sequence, human phenotype, and known properties of molecular target mechanism. And they then prospectively predicted what drugs were currently in phase 3 would be successful, i.e. approved by the FDA, and which ones would not. Okay, this is 2009, based on their results from machine learning. Now, there's a small n here, as you can see. But the ones, so these are the ones they said were promising. These are the ones that say were not promising, based on machine learning of a whole bunch of variables. They were pretty good at predicting which would be approved by the FDA, and also pretty good at showing that drugs that were in phase 3 would go backwards in terms of going to an earlier trial or discontinued altogether.
So Jerry, there was a lot of different data.
Yeah, they had a lot of different variables.
Like a ton of data, and they were looking for,
They used to support vector machines. Actually, they used a series of them.
Just looking for, hidden variables using the strong specters and stuff like that.
But I mean, the N is quite small, and I think those like Alex Linen back there would probably take a lot of issue with this paper.
So the
it's
other
it's
thing is
interesting.
that so the data that was collected in phase 1 or phase 2, like the tox data or something, it was all available.
It's all available. Yep.
So yeah, so it's not like, I guess.
Well, this is a
It's
important, interesting
not like
point.
it's going to replace all the work in phase 1 or phase 2. It's in silico.
No, this is interesting because, well, I'm getting there though.
Okay, go ahead.
You want to finish?
Jerry, may I ask a question?
Yeah.
Given that some of what's being talked about here is similarity to existing therapies, it might be true from a regulatory perspective that things that are more similar to existing
Just read the title of the
therapies.
slide. Drugs similar to those previously approved by the FDA based on molecular mechanism, clinical interaction, target pairs are more likely to be approved by the FDA.
I agree, I'm saying that, but I'm saying given that this is the case, isn't it likely that to some extent the differentiability, the predictive power of what will be approved is largely based on similarity as opposed to what might be effective medically?
Well, yeah, except that the role of people on advisory boards for the FDA is to look for things like adverse drug events and efficacy. And now, there's of course, I don't want to go into the politics of this because it becomes very thorny very quickly. Okay, but the major support, I guess everyone knows in here that the U.S. Federal Drug Administration is largely supported by its sponsors. You know this?
Yeah.
And who are its sponsors? Do you know?
Big Pharma.
Big Pharma, right? They account now for about 92 to 93% of their budget. So
that was PDUFA that created it.
Yeah.
Probably PDUFA.
Yeah. So I'm not implying anything or trying to say anything, but anyway. So this is just showing for validated pairs, that's molecular mechanism, clinical interaction, target pairs, as they've described. And this is the percentage of the total of the 880 currently FDA-approved drugs, which have been actually approved historically. The majority are based on previously validated mechanism indication pairs. Okay, does that make sense? I don't mean, is that the right thing to do?
So
I'm
does
just
that
saying.
like mean they're kind of Me too. I mean,
Yeah, they're all like that.
Yeah.
Well, not all. I mean, these guys aren't, but the majority of ones that are novel, not based on these, fail. And I'm not going to show that data or those data. So this is the second challenge, which, and I apologize, a lot of text here, but if it's true that molecular mechanism and clinical indications, could be paired from well-characterized patient cohorts, then wouldn't this provide the ideal sort of training set if you're using deep learning or machine learning? You see what I mean? And the problem is, how do you gather this data? And I've already alluded to that. So, here's a statement from Eric Lai from Takeda. Electronic health record and other biomedical data are contained in repository silos, exist in different file formats, are distributed among various clinical environments with different cultures, and data cleansing often results in data degradation. So anyone that's ever worked with electronic health records sort of knows about this, right, Brian? Anything? I know there's.
Well, I think I was sitting next to Eric when he made that statement.
Okay. Ansha, this is amazing to me. So noted in December that 227 million Americans, or 69% of the U.S. population, had lost protected health information contained in their electronic health records. This is an exact statement. I can give you a link. This is obviously a challenge and remains a legal problem for hospital administrators, but there is an emerging concern about clinical IT professionals that these data cannot be protected even when using applications such as blockchain. You know what blockchain is?
How many people know what blockchain is?
Sort of.
I'll tell you what.
So really, you guys
that Bitcoin did took care of that for you.
Okay. That's what I'm hoping. And then we go back to the partnerships between Big Pharma, UK, Biobank, Finland, Estonia, Poland, and China, where there are fewer restrictions on patient data, but it's sort of ironic that, this does not mean that the entire electronic health record of 69% of Americans, but that some of the protected health information. We should go on the dark web, though it's real slow, I've been told. This is from Quintiles, IMS Health. Of course, they sell this stuff. Vendors of data analytics will make access to patient data a mandate, not a competitive advantage. So many companies now are directly selling comprehensive but aggregated de-identified patient data linked to SNPs and genome sequence. And these are some examples. And I like this statement here, although it's from Frost and Sullivan, which is one of these marketing or, forecast groups. A lot of healthcare products and solutions are focused on one or more steps of a patient's journey. However, companies like Prognos, which is in oncology, look at the whole longitudinal care pathway, from prevention, prediction, diagnosis, treatment, to management of the patient. And that's, I think, something that's coming,
Oh,
and bunches of diseases. So then I found a picture of them because there were so many text slides. I thought I got to liven some people up that have already fallen asleep. So this is from last year's Biodata West. That's Vern DiBiase, who's head of digital data and analytics platforms for GlaxoSmithKline. The real challenge is not a lack of data scientists per se in drug discovery and development, but a lack of data curators who can prepare and integrate multivariate biomedical data from disparate sources for experimental pharmacologists and software engineers. Now, I thought that was an interesting statement because a lot of people I talked to at that meeting from pharma told me exactly the same thing. It's not that anyway. So now we come to the scarce item, which is the blue unicorn. If you've heard of this, which is that Big Pharma cannot find skilled AI researchers. They're looking for the blue unicorn. There are very few scientists who are both experimental pharmacologists, including, subdomains of things like medicinal chemistry and such, who are also computer software engineers with skills in deep learning or programming. So this is kind of looking for the blue unicorn, which is a sort of a cliche. So Big Pharma has gone to workarounds. One is to train in-house experimental pharmacologists in computer science. I can tell them that's probably not going to be successful. That's my own bias. But cultivate data curators, like from the last slide, from the existing pool of scientists to act as an interface between experimental pharmacologists and computer scientists. In other words, people who prepare the data, try to get it into a format that others who are biologists, who have some sort of skill, maybe in R and maybe a tiny bit of Python, but usually not, can actually deal with it. And then finally, to outsource or form partnerships with deep learning companies. So, you know, this is interesting because they were all saying this to me. People from Merck, from GSK, from Roche, Genentech. So, you know, they're sort of trying to bridge this divide, which is not really data science per se, or maybe it's a variant of it. You know what I mean? But I think our students who are really good programmers and can use deep learning methodology are going to be very successful if they apply at this point.
We do have some pharmacology classes that some of you can take. You know, we have one that I've been involved in, 601 and 6202. Translational pharmacology is one that we put together with Donna Swetek years ago. So we do, you know, and we're looking at that. We've got a couple students that are in the pool of applicants now that could bridge this. But it's basically you need some pharma experience and bioinformatics.
Yeah, I don't.
This is actually a pretty good pathway for students who want to work in pharma.
Yeah, because there are a lot of jobs there, right?
Be brought up, you know, instant jobs.
Yeah.
I think.
So anyway, I thought that would be interesting, and I know this isn't really tool and tech like, but it's going to get even less like that. So this is the competitive space and growth rate part of the talk. So these are the top vendors here, identified by their logos, that bundle that. Have you ever, anyone here heard of real world evidence? That phrase?
Yeah, this is an important one, and I'm not seeing too. This is actually a serious buzzword in clinical research. Right?
It is, and I see this. I'm saying, what, you know, does that mean truth? I don't know quite what that means, but anyway, what they mean, I think, is that they can get patient data and along with some genomic or expression omic data, and in aggregate bundle these and include them with their analytic platforms, like Cerner, who's a big electronic health record company, Blue Health Intelligence, which, by the way, if you, like me, covered by Blue Cross Blue Shield, they're probably taking all of your data and selling it to other people. I don't know if you have to sign some form, but I think I had to, and I didn't even read the stuff. It's too long. Anyway, so these guys do that, including IBM Watson. Now, this may not be of any interest to you, but if you're going to go out in the world and find a job, you've got to realize that a lot of these companies are companies that you could probably get jobs with.
Well, I'll put it this way, that I've had many students who've come to me kind of at the end, and they're actually interviewing with these kind of folks. And, you know, they want like a quick kind of mind meld. What, you know, gee, what kind of question might I have? I mean, we had several students who have gone this way.
Right, that's good. I mean, well.
So it's really good to know about.
This is the I know this from other people, but it's also on this. I can give you a link. This is currently what these pharmaceutical companies, J&J, I think, is the largest now in the world, or Pfizer is. I can never remember. But these are, you know, J&J, Johnson & Johnson, and especially Janssen, which is a division of it, GlaxoSmithKline, Sanofi, Roche, Genentech, and Pfizer. And these are their major partners for doing artificial intelligence for drug discovery.
Interesting. So they've all kind of stratified this.
Yeah, this is benevolent. That's a funny name, AI. I guess they want to get rid of that notion that Elon Musk propagated of these robots running down the street killing people.
That makes me trust them.
Yeah. There's Alex Kalinin, friend, isn't he? Your buddy is in silico medicine. Both these companies partner with GSK. This company partners with, these are all the primary partners. So Sanofi, GNS Healthcare, which I know well from Cambridge, Mass, is partnering with Roche Genentech to do AI for drug discovery. and IBM Watson Health with Pfizer.
Hey, Jerry, are these deep learning companies independent companies, or are they ones that have now been subsumed by their partners? Like, is
They
Benevolent
haven't
actually
as
owned by?
of the end of January 2018. And I mean, I know the people that are the executives of this company pretty well. And but I have never even heard of this company. But anyway, so I know that GNS hasn't been acquired by anyone, but I'm sure they're making themselves targets for acquisition, right? If this ever works. I mean, that's the other thing I haven't said.
This is really interesting. So what you're
Well, not IBM probably.
What
but
you're saying is that the various drug companies are lining up with partners.
yeah, and there are tons of these startups in artificial intelligence in the sort of biomedical arena, you know, doing all these different things. Certainly deep learning for computer vision, right? And radiology and ophthalmology and pathology, which have been very successful. There are also a huge number of them in drug discovery. So it's kind of interesting to see they're sort of sorting out along these lines. I'm going to go pretty quickly. And these data probably are not to be believed because they're forecasts from companies who are paid to sell these market research reports. Frost and Sullivan, compound annual growth rate of AI in healthcare from 2015 to 2020, 15% growth. And you can see revenue and the growth rate there. Some of this is obviously forecast per year.
Wow.
CAGR is compound annual growth rate. This is from Coherent Market Insights or Incoherent Market Insights. Value by end user deep learning in drug discovery and diagnostics. And this is global. Pharma companies, which account for the majority of this. And this y-axis is weird because it's in US millions. So 250 million, 1.5 billion.
Okay.
Y-axis. So, I mean, you should take this with a grain of salt, but I've got to tell you, and others who ever follow biotech commercially, this space has been tremendously hot for about a year now. I mean, hotter than the, Wall Street index would you believe. It's been incredibly hot for investment. So, it'd be interesting to see what happens. This is getting back to Brian Athey's question, sort of what McKinsey, MIT, and I call them that because all the people I know at MIT, even at the Broad, seem to work closely with McKinsey, so they must have some kind of contract or preferred contractor status. This is about the trajectory of AI-related technology. I think this was published in 2016. It's weird because they have these phases. They call them the ecosystem, but I actually deleted that from the slide because I have a very specific, I grew up sort of being a field biologist early on, and I don't like people who use the word ecosystem when it's not talking about actual ecological system. Anyway. So I just call it phases. Creation, survival, growth, equilibrium, decline. Then these weird, they look like buttons you would push, right?
Yeah.
Fast forward. Less than a year, 1 to 3 years, 3 to 5 years, 5 to 10 years, over 10 years. And then they talk about, the only thing that's on the red line is patient swarm technology, which I don't even know what that is, but anyway, my apologies. But if you look, you can see here deep learning platforms for drug discovery, and they say 3 to 5 years from 2016. That's on this kind of trajectory, growth, although I think it actually occurred earlier, but they have natural language processing, obviously another thing that can be done by machine learning.
I got to say on this natural language processing, I had dinner with this fascinating guy last night, a fellow named Santiago Segura, who's a data scientist we're trying to recruit into ECE and Midas. And he's an incredible guy, young guy, and he wrote a paper using nearest neighbor word correlations.
Oh, really?
Comparing Christopher Marlowe's work, like Dr. Faustus, with some of William Shakespeare's work, and proved that Henry IV was a play that was written collaboratively by William Shakespeare and Christopher Marlowe, published in the latest edition of the Shakespeare Quarterly.
Wow.
So I was trying to get him interested in looking at diagnosis drug adverse event pairs in our electronic health record, of which we are accumulating 1.3 million new documents a day, transcribed notes into our electronic healthcare record from either the transcription that we do here or referrals, which interestingly and sadly are still coming through our system through paper and fax.
is probably
like
the
that.
fax.
So
you got to get rid of those.
I would say 85% of our electronic medical record is free text.
Oh, really?
Yeah.
Wow.
And so
That's
this
terrible.
natural language processing, I think it's got to be real big. Yeah, that would be one that we would watch. Wearables, I mean, that line up there. AI optimized hardware. This is incredible.
Yeah,
And, you know,
there's just a nature article. I
it's
thought it was the
just
last line of this
happening.
talk.
Yeah, so I'll let you make that point, but that's something to really watch. And the big problem there, of course, is that it's all a black box, and the physicians are going to be walking around with this damn stuff. We had that talk the other day, and but it's coming, and it's in the chipsets that are coming from Intel and this kind of thing.
So I don't know. I don't really know if they know anything about what they're forecasting. Anyway, there's some interesting things. A diagnostic decision support.
I think there's no
up here.
question about it. Yeah.
I mean, geez. Anyway, yeah, my son is a graduate student in CSE, computer science engineering here, and he took a course in natural language processing, which he didn't like. But anyway.
Well, one thing I got to tell you. So I gave a keynote in opposite the health minister of China in Hangzhou right after the G20, and the first question from the audience, and when I've given the talk in other places in China, which I found out yesterday at dinner, it appears as if China is in the process of collecting blood from every patient that walks into a hospital and sending up to Shenzhen and BGI, the entire country. And when I gave this talk, and this was to 1,200 hospital executives throughout China, it's real-time translation. It was kind of a G20. The first question that came from the audience, and the question I always get in China is the computer going to replace the doctor in 5 years? Are we getting rid of doctors?
5 years?
Yeah, 5-10 years. I mean, they wanted to know. And they were serious. It was
Well, we should close
so
on
I'm
the
standing there and I'm
medical
like,
school
God, I
training
really
program.
never thought of it. It's like, what are you going to say? And of course, you tell them no. But if you look at like this diagnostic decision support and some of these things over there, what the doctor and the care provider more generally does is going to change significantly. And we don't actually know how that's going to play out right now, I think.
Who will be liable?
Well, by the way, and that's
the software maker or
I
whatever.
think that's a really important question.
TensorFlow.
It's a very important question, and that drives a lot about defensive medicine. So I was in a meeting in Nashville a couple weeks ago, and I shut up in just a sec here.
That's fine.
And we were looking at a product that could detect drugs in your plasma, with small drops of plasma. And we were saying that there's certain drugs that you simply have to be off of before you get anesthesia and surgery.
Yeah, right.
And so the thought was that we could get that product right into the pipeline with the anesthesiologists and the surgeons, because there's the payers would just, there's such a threat of lawsuits because the patients lie, the patients forget, the patients take a bunch of drugs that they're not being prescribed, they're on opioids or whatever, and if they're on them and they go under, need the anesthesia, they're going to die. So this defensive medicine thing is absolutely real, and it's related to that. And it's going to drive a lot of this. Most likely it's going to drive more devices into the hands of physicians and caregivers.
But they're not going to be prepared for them unless you
Yeah.
train them now how to use them, right?
Right.
Anyway, a couple of examples, and I'm not going this into detail. This is from, again, Jeff's Google Brain Group. What does a molecule look like to a machine? This is important because he's looking at quantum molecular dynamics.
Wow.
And these are very good links. I'm not going to go into this in any detail right now, because I don't have any time. But I find it fascinating. It's related to things you and I have recently talked about, Brian. And the basic idea in this publication is to use a different kind of neural net model to approximately, to reduce computation time as compared to using other methods. So it's a message passing neural network that predicts the quantum properties of an organic molecule by modeling computationally expensive DFT calculations.
Two very important.
This is something we did with Vikash. Treatment resistant to mood disorders. This is, and Brian, I apologize for presenting this. This is 127 nominal variables from 3,017 patients. These are examples. 25 SNPs. These are some of the different kinds of data that were used. And this is the BaseDB outcome. This is using probable computing with BaseDB. What would be most predictive? One being completely predictive of treatment resistance to mood stabilizers. Here are some SNPs and here are some things from the electronic health record. That's the guy, really smart guy, Vikash Masinga. And finally, to end up with, Alex isn't looking, Alex K in the back.
Oh, yeah.
I really find this article to be very good.
Alex, that's our latest and greatest out of our
No,
lab.
it's not this. It's the one that you wrote with a whole bunch of people from one of them from the University of Pennsylvania.
Oh, this is a different one?
Yeah, I took the heart of it out where somebody, I don't think it was you, was talking about applications for drug discovery like
Alex, I didn't
ligand
know about
prediction
this
and stuff like that. Ligand receptor prediction.
Isn't this the one we just finished?
No.
This is a different one?
That's the older one.
Has this ever been published anywhere or just on bioarchive?
It's revised.
Okay.
Oh, I see. Yeah, no, we did pharmacogenomics.
Yeah, this is quite a good
Pharmacology
review though,
stuff.
especially for like deep learning and drug discovery. It goes into a lot of detail. So now I want to get out of this, but anyway, I'm giving, oh, good gracious, supposed to be giving at the end of this month a workshop on AI and drug discovery. This horrible picture of me. So I'm trying to find someone who will take my place, RA or Alex or Brian, or anyone else.
Sure.
Okay, sure, says one person.
Careful what you wish for.
I'll wear the nose and glasses and pretend to be Gerry.
And a fake beard,
Yeah.
And they want me to now co-chair the conference. This is one of those handsome wave things, Brian,
You gotta watch out for this.
Yeah, they were insidious. And then this one, which is in March, which I do actually want to go to, but I don't necessarily want to present, which is Biodata World West, which was actually a Google UCSF combo. And now it's also involving NASA Ames out in San Francisco. I find this conference to be really, really good.
So you better get them to change that to research professor so that not.
Yeah, I know. I knew you'd see that. I was going to.
Better fix it.
Yeah, well,
Well, these are
I can change it to
opportunities,
janitor,
but
that's fine,
you
too,
can
yeah.
see that
and this last slide. This is from the latest Nature. Big data needs a hardware revolution. Were you just talking about this?
I was. I mean, in fact, I want to mention this to the group. There's a fellow named Satish. Don't ask me his last name, I'll butcher it. He's a professor of computer sciences that's been coming to dinners with me to recruit faculty in Midas. And really his interest is new hardware configurations and architectures to accelerate, computing for bio applications, genomics and so on. Marcus, he's a cool guy. I really like him. He's an intellectual.
It's like bio-inspired chip design.
And I want him to come visit. I thought, Alex, you'd be great to meet on your pipeline. I think you can speed it up. I want him to meet Yang Zhang. He has methods and architectures and approaches to giving you a factor of 1,000 compute enhancement, that kind of stuff, which could take something that would be could take something that's very difficult and because it takes a few months, like that. I really thought, to me, the thing that I was excited to see, Gerry, was that really neat slide you had with the deep learning and the molecular structures.
Oh yeah.
Because I think
originally that was like 12 slides I had, and I thought, no, I'm going to.
I think that's really a fantastic thing because I can definitely see. We were talking about this last night at dinner a little bit. I mean, deep learning. For guys like me who used to live in Fourier space for 15 years. I mean, you can see when they're looking at those cats, you can just see it's Fourier decomposition and convolution 101. I mean, you can see all the spatial, but you can see it. Yeah, similarly, with these, because what I was excited about is if you go back to that one slide that you had You know, where you had that great timeline of all the different kinds of research that's being
Yeah, that's
done.
way back here.
My sense is what's going to happen is this. I mean, looking in the crystal
Wait,
ball.
hold on.
Yeah, keep going.
This one?
That one. See, this so-called rational drug design.
Nah. Yeah.
Okay, with the small compounds. That's the way we used to work. Heather Carlson down in the pharmacy school. You know, we had a couple of our students got PhDs on MOAD, her mother of all databases. I mean, Nick Kozanoff got one.
DoD stole it for mother of all bombs.
Doug Benson, some of our earlier students got. You know, there was a lot of grants, big center grant from NIGMS. And a lot of it was just having to do with ligand binding and pockets and all that kind of stuff. But, you know, if you took that kind of approach, like, for example, this mother of all databases, we have all these ligands and the three-dimensional structures, and we have kind of the three-dimensional structures of all these catalytic binding sites. If you coded that up, you know, and process that with that deep learning thing, you know, those vectors would resolve just like those pictures.
Yeah, they do.
And you could jam through that whole database and like nothing flat. And you could really see how these drugs were binding into those pockets or not and that kind of thing. I mean, so what I'm saying is that some of that earlier stuff is going to get transformed by AI into new stuff.
Yeah,
I can definitely see that.
Yeah, this is going to jump like that.
Yeah, it's going to jump.
Yeah.
Because these pharma companies are still completely wedded to these old technologies, and they're looking for new ways to kind of
And most approved drugs are like, you know,
I mean, they're chemists.
they're similar to other drugs that have been approved by the FDA, right?
So that, I think this chart is really worth looking at a lot. And, you know, the biggie, you know, and I don't know. Now, China doesn't have China does, thank goodness for them. They don't have EPIC, CERN. They have a lot of homemade EMRs over there, and so it's still early days.
Oh, that's good.
I think it's really good. There's a lot of it. So I think what will happen, what happened in America was that when the ONCHIT, the Accountable Care Act came in 2009, we had all the best intentions in the world. And Chuck Friedman, who's chair of the Learning Health Sciences, was second in command of that whole thing. And he was the policy man. What we wanted was, we were at 20% adoption of the EMRs in the country. It was awful. You know, we got airline industry and all this stuff is fully digitized. Healthcare was in the dark ages. So we said, we'll legislate, we'll induce people with incentives and then penalties later on to switch. Our problem was we only had like 2 or 3 choices. So we created inadvertently monopolies. EPIC, Cerner, Allscripts.
Yep.
You know, they became monopolies. That's not. But in China, I think there's so many different.
Yeah.
Well, they're going to let the market play, and then from the top down, the government will start killing them off in various ways, and you'll end up with a monopoly, but you'll have a damn good EMR.
Yeah.
That's what's going to happen.
I hope that's what happens.
I'm quite convinced of it. And BGI will be right in the middle of it. There's no question about that. So, see, because one thing Gerry kind of didn't say, but it's certainly true, the reason that so much of it's going offshore is because there's been a lot of studies for patients to go through the trials here. They cost about $54,000 a patient to take them through the trials, phase 1, phase 2, phase 3.
I didn't say that.
You know, so if you're doing a randomized control trial with a big N, you know, it's the clinical research study costs you $100 million. So if you go to India or you go to China, you know, you can get the price by a factor of 20.
And by the way, a lot of randomized controlled trials, phase 3, actually have small n's.
Reflects yours.
Well, that's a problem. However, we thought about that a little bit. Han Chinese, right? It's 95% they're Han. But, you know, they still are going to have different variants, and they will respond to drugs in different ways. You know, and actually, in some ways, it's going to make it easier in the early days to do some of this stuff.
Yeah.
You know, I don't know. I was asking Steve. He's gone. Mark McCarthy's talk that he gave. How many people? Did anybody see that? Yeah. What did you think, Ricardo? It was awesome. It was awesome.
Yeah, we had lunch with him too.
Oh, did you?
Yeah.
You, especially on this palette stratification of various kinds of type 2 diabetes.
That's
it was really amazing. We're going to try to get that PowerPoint from Steve that we can send around, but I think that would be really, really worth looking at. You know, and I must say that he's taking some of these. They're taking some of these approaches here.
That's why I'm I'm working also on this thing that we've talked about. You know what I'm talking about.
All right,
Okay, was that okay?
I thought it was great.
It wasn't very technical.
Thank you.
But,
There's a lot of potential for good careers here.
yeah, that's for sure.
That's the point.